So now that we know it was a biogenesis theory which was actual and is justified. To create a living thing, there must be a living source from any non-living particle or any non-living things like decaying meat or some rotten wood. No microorganism or living particle can grow. And it is Louis Pasteur who finally claimed with his short night flask experiment the clarification of biogenesis theory. Now let's see what is scientific method. The scientific method is a generalized framework for conducting an investigation. So all the experiments we have seen that the debate over two theories and how they approach or design some experiments. So all the experiments or all the approaches they have taken in order to claim something, to hypothesize something, and to make sure that yes, it is right, it is justified. Scientific methods are important. It is proved scientifically. So what is it needed? What are the facts and important factors of a scientific method, which is a group of observations leads a scientist to ask a question about some phenomena. The scientist generates a hypothesis, a potential answer, or explanation about something. Just we have seen in the debate. There they first observe a phenomena and ask questions that whether and how the microorganism arise, whether from a non-living source or a living source. Then they generate a hypothesis, like two hypotheses, one spontaneous generation theory, another is biogenesis theory, and then they conduct experiment in order to clarify their hypothesis. The scientist designs and conducts an experiment to test the hypothesis. I just said this. So based on the observed results, the scientist either accepts, rejects, or modifies the hypothesis. So based on the experimental results, either they can accept that yes, like Pasteur, yes, it is right and it is acceptable in the Royal Society or in a scientific society, or they can reject it, just like the spontaneous generation theory, that there is no base of this theory, so it should be rejected. Or they can modify it, right? They can change their experimental approach or their methods, their sample number, everything they can change or modify in order to claim their hypothesis. And finally makes a theory. Some definitions. What is hypothesis? I'm claiming this is hypothesis, this is experiment. Now what are the definitions of this? A hypothesis means a proposed explanation for a set of observation. That means you have observed a set of phenomena, and based on these you have claimed a thing that yes, based on the phenomenon or based on these incidents, I can say that this is right or this is wrong or this is the way. These are hypotheses. An experiment means a test using controlled observations to confirm or refute a hypothesis. It is simply a test with some sample number or some sample or controlled observations in order to either confirm that hypothesis or refute that. Either you will go for it or go against it, right? This is experiment. And finally, what is theory? It is a long-standing hypothesis. A theory is basically a hypothesis which is standing for a long time or which is persisting for a long time, repeatedly verified. That means so many scientists work on it, so many scientists work on this hypothesis in order to purify it, in order to clarify it, or having a final justified result. Right, so repeatedly verified by experiments that explains a wide range of related phenomena. What caused fermentation? You might have remembered that how microorganism has arisen. There were 4 different questions, and it is the second or third question, right? What causes the fermentation? Well, fermentation refers to the formation of alcohol or lactic acid from sugar. Pasteur's experiment observed that yeast cells only come from other yeast cells. Demonstrated that yeast can grow with or without oxygen, that is, they are facultative anaerobes. Pasteur first claimed that yeast is a fungus that must grow from another yeast or another fungus. And yeast is an anaerobic fungus. It doesn't require any oxygen or any air in order to survive. Showed that bacteria ferment grape juice into lactic acid while yeast ferment the juice into alcohol. So the 2 different types of microorganism, both of them work on grape juice, right? Both of them work on grape juice but produce 2 different types of products. Bacteria work on grape juice in order to produce acid, and yeast to produce it alcohol. And by learning chemistry or organic chemistry from your intermediate life or in honors level, I don't know whether you have go to that organic chemistry course or not, but you know what are the basic difference between an alcohol and acid, right? 2 are basically or totally different things. and two microbes generate totally two different products. And finally, Pasteur invented pasteurization. Pasteurization is a procedure to apply heat in order to remove some harmful microorganism from a food source. As we know that we all drink milk which is pasteurized. That means the harmful microorganism from a milk has been eradicated or removed by applying heat at a certain amount. Buchner's experiment showed that enzymes in cell extract cause fermentation. That means he first showed that enzyme in cell extract, it is an enzyme from microorganism or microbial cells that finally cause fermentation to occur. Initiated the field of biochemistry and the study of metabolism. Well, Buchner's experiments or the role of the scientist is very, very important. He initiated the field of biochemistry, which is now the mother of all scientific study, biological study. The final question, what caused disease? Not the final, the one before the final. What causes disease? Regarding this answer, to find out this answer, the scientist who worked on it is Robert Koch. The germ theory. He explained a theory which is called the germ theory of disease, states that microorganism can cause disease. In this theory, Robert Koch explains that it is microorganism that may be responsible for happening any disease. He demonstrated the relationship between a microorganism and a disease. He find out the relation, like how microorganisms are related to cause a disease. He demonstrated that mechanism or that link. He postulates a step of experimental design in order to justify that yes, microorganisms are responsible for causing a disease. This is called Koch postulate. Koch postulates are a series of steps to show that specific microorganisms cause a particular disease. Koch and his colleagues significantly advanced microbiology lab techniques. How can we prevent infection and disease? As we know that due to some microorganism or microbial infection, people generate or develop some disease. So how can we prevent them? How can we prevent the infection or to inhibit the disease to cause? Several ways: hand washing to reduce infection. Joseph Lister said, developed aseptic techniques including phenol solution to treat bandages and surgical instruments, which has significantly reduced post-surgical infections. Ehrlich's magic bullet led to field of chemotherapy. Here you can see the name of some different scientists. Semmelweis is one scientist who said that hand washing to reduce the infection. Now what we are listening this after so many years is yes, hand washing can prevent us from developing coronavirus disease or COVID-19. And Joseph Lister, his applications were in the field of medical microbiology, where he says that if it is infected certain instrument after surgery, then it will reduce the number of post-surgical infection. Then Paul Ehrlich, another scientist, you can see here, who invented the method of chemotherapy, which is the most important treatment for cancer biology or cancer patients. Edward Jenner, vaccination. He discovered vaccination, the father of vaccinology. Edward Jenner, he said the smallpox vaccine led to a field of immunology. Nightingale's antiseptic techniques and nursing practices led to the field of nursing. So these scientists have impact in different fields. Paul Ehrlich in case of chemotherapy, Jenner in case of vaccinology and immunology, Nightingale in case of nursing. Snow studies of cholera epidemic in London started epidemiology. So he started the study of epidemiology, the study of epidemic, the study of occurrence, distribution and spread of disease in population. That means how a disease is spread from one population to another. Now we see that the SARS-CoV-2 has been transmitted from China to USA or Latin America or even in Bangladesh, everywhere. This is called pandemic, or this distribution, how this disease is being distributed or this virus is being distributed all over the world is the study of, is a field called epidemiology. Now let's see what is the modern age of microbiology, which is examining the basic chemical reactions of life, determining how genes work. So modern microbiology totally confined within the genetic knowledge, right? Recombinant DNA technology, genetic engineering, microbial modification. That means determine how genes work using genetics, molecular biology and recombinant DNA technology, determining the role of microorganisms that play in the environment. So that is all for your first lecture. Hope to see you in the next one. Thank you very much.